Okay, so I'm Sunil Nimithy, and so my project was the FastMapper, so the mapping of reads, and I'm doing the hard level. So I'm a graduate student here in computer science, and I actually started out at UCSB doing biochemistry, and so I was doing X-ray crystallography in the lab that I worked at. So you get these, from the X-ray crystallography, you actually get these electrostatic maps from which you can determine the three-dimensional structure of the protein itself. But you need to know the sequence of the DNA in order to do this. So it's really important that we find a way to efficiently map the DNA reads that we get, these short reads, to the DNA sequence. Because if you have any sort of mutations in your DNA, it can affect the structure of the protein. So for example, if you have sickle cell anemia, you're going to be having, the protein is actually going to have different properties. And so the problem is that traditional methods for doing the short read mapping are inadequate because they simply take too long, and the rate at which we're generating these DNA reads, sequencing is becoming more common. The rate at which this data is growing is increasing. So we want to find a more efficient way in which to do this. So my solution or my analysis was to look at 3 different ways in which we can do this. The first was just using the brute force trivial method, and that's the sliding method that everyone else has been looking at. The next was the hash map that was covered briefly in the previous topic, as well as the Burrows-Wheeler index, which we've seen a couple of times. My benchmarks were accuracy, throughput, and memory use. And so the sliding algorithm we've seen a couple of times. It'll just move your read along the DNA. So you'd move this all along the entire genome. So you're actually looking at a linear time algorithm. And you're going to find the place in which it has the the lowest number of mismatches. A hash map algorithm, I'll just make this a bit bigger. So what you're doing is you're actually taking your short read, and you're going to be chunking it up into smaller reads. And you're going to be matching or hashing these and finding the locations at which they match your genome. So what you're doing as you slide along, you're going to see that it maps in these different areas. And then when you kind of rotate along based off of the offset of your read, you're going to find that you're going to get your reads are actually going to align in certain places. And depending on where the reads align most, that's where you can say that this is where your read is. The Burrows-Wheeler index we looked at before, so this is the same string. It's gigantic. When you're looking at the full 3 billion base pairs, this is absolutely enormous. And so what you're going to be doing is you have your last row, which is your FM index. And by going through, I mean, we looked at this other time, so I can kind of skip around. But by going through, you can find, you can iteratively walk left and find the way that it matches. But what I wanted to look at is how we index this last string here. So we didn't really go over this in class, but you're going to be, the way in which you index that string, how you, is going to have a big impact on your performance. So my testing was done in 2 parts. First part used random data so that we can determine the effects of the size of your genome on your performance. And the second part was actually using the human genome 19 build 37 in order to validate this. And this actually gives some important, that has some important implications. Like we looked at with the last one, is that using simulated data gives you a bit nicer results than using real-world data. So the input was the human genome, or the genome itself. Reads were generated from that genome. Because I really wanted to stress the algorithm a lot, there's significantly more errors per read generated from what I was using than you'd find in the actual human genome. So there's approximately 100 times the amount of SNPs that you'd find there. And the output is the SNP itself as well as the location. So you're either going to have 0, 1, or 2 SNPs as well as the location that they occurred in. So we didn't really go over in class how you build this FM index. Christian just talked last time about how you keep basically pointers to the index in the genome. My implementation did essentially merge sort and then quick sort with the individual. So what you do is, by looking, you can sort the A's first, or and then the C's, the G's, the T's, and then merge those together using a quick sort underneath to sort those. If you have less RAM in your system, you could sort the AAs or the AAAs and continue on that way. The less you divide it up, the faster it's going to be, though. And really, the amount of time it takes to compute this index doesn't really matter because you can store this and you never have to do it again, assuming you're using the same reference genome. So the test setup, I actually have a ton of RAM. This is an old server that I bought off Craigslist for cheap. So I was actually able to test the hash map using the full human genome. So starting with the space comparison, so if you are actually doing the whole hash map, you're going to be consuming a lot of RAM. So for each base, you have an integer, a 32-bit integer. So it's going to take over, about 12 gigabytes of RAM. Burroughs Wheeler, we capped that at 5 gigabytes, so that was the hard project, was to be able to get the FastMapper to run in 5 gigabytes or less. You can decrease the frequency at which you index that string in order to decrease your memory use. And lastly, the trivial uses a small amount of memory because it only has to keep the length itself, the genome itself. So going back to indexing this Burroughs-Wheeler string, we gave 5 gigabytes in total in which to index this. But when you walk left, you need to know the count of the characters above you, the A's, C's, G's, and T's. And when you want to determine your position in the genome, you're going to need to have references to, well, this maps to position 5 million in the genome. So you can separately index the position as well as the counts. And to make this a bit bigger, so by changing the rate at which you index where it is in the genome, so this first character here, so 3 means that every third position has been indexed for where its location is, versus the number of counts, so every 77 times we index, there's this many A's above us, this many C's above us, etc. So what you find is that you really want to focus on, to get the most performance, you want to index the location much more frequently than the counts. So actually using 3 and 77 here will give you the best performance while maintaining a 5 gigabyte size. This is the same thing, but now this is using the human genome, so you can still see. And by the way, these are log graphs, so you're seeing as the genome size increases all the way up to 3 billion. This is the performance. So now in terms of throughput, the trivial algorithm just kind of gets hammered. So as the length of the genome increases logarithmically, you have this decrease in the performance of the trivial algorithm. And so you can see in the human genome runs, the hash map and the Burroughs-Wheeler just completely destroy the trivial algorithm. It's not even really visible out here. So both of those are much better than the trivial. But you can see the hash map, which is somewhat expected to be performing better than the Burroughs-Wheeler, is still outperforming it. One of the reasons is that we talked about the trivial algorithm being this linear run through. So you have a huge standard deviation. As the genome increases, you might find a match in the beginning of the genome. You might find a match all the way at the very end of the genome. So you're going to have a huge amount of variability. The hash map has the lowest amount of variability because it's a series of operations which are all order 1. So we're just taking a bunch of hash lookups. And the Burroughs-Wheeler will depend on a number of things, but you'll find that when you look at actually the human genome, this is a bit larger of a difference than you'd find here, just because real data itself will have repeats and things like that that you don't have when you do simulated data. So it's going to be a bit harder on the algorithm. In terms of accuracy, they both, the hash map and the Burroughs-Wheeler, fare fairly well. I mean, not compared to the trivial. The trivial does the best. Again, this is having 100 times more errors than you'd find, or SNPs than you'd find in the human genome. So the accuracy is pretty low. If you run it with the 1 in 1,000 that you normally find, the accuracy of all of them is near perfect, just because we're not looking at insertions, we're not looking at inversions, we're not looking at a lot of things that you really find, so it's kind of an unfair comparison to just include a 1 in 1,000 error rate. when I ran it, I was getting 3 errors in 1,000, which is pretty insignificant. But you can see here that between simulated data and real data, again, you're getting a big amount of difference in terms of the performance of your algorithm. But the most important part is your performance per megabyte. So if you were tasked with building a system that was going to handle DNA sequencing, and you're going to have multiple machines, assuming you're Illumina or something like that, you might actually want to look at using a hash map as opposed to a series of relatively low-powered machines, because you can get better performance per megabyte by doing a hash map implementation. Admittedly, my Burroughs-Wheeler performance was not as good as Bowtie, but I mean, I wasn't really expecting to beat it. So potentially, there's quite a bit of improvement that can be made on here. So the key points to take away is that you want to make sure that you're separating out the indices when you're doing your FM index. So in my case, if you want to bring it down to 5 gigabytes, 3 and 77 would be good benchmarks to start out with. The trivial algorithm is going to be the most accurate, even when you scale out to all the way out to 3 billion. It maintains a high level of accuracy. The smallest amount of memory you can run in is this Burroughs-Wheeler, while still maintaining an appropriate amount of throughput. HashMap is giving us the best performance as well as the best performance per megabyte, but it does require a system that has enough memory to run all that. And then finally, the simulated data, you can use that to get, landmarks and to study the trends. But until you throw real data at it, you're going to find that you're going to find a lot of surprises when you finally do throw real data at it, because your algorithm is going to kind of hiccup a bit under some of the stress of that. Future work, you can always make this out into multiple threads. Going into CUDA program would be interesting. So I wrote this all in C because I planned to port this over to CUDA, but kind of ran out of time. You could actually increase the compression on the Burroughs-Wheeler string, so you find that there's a lot of repetition that you can actually take advantage of and get this significantly below where I had it. So that could actually, again, throw off the performance per megabyte. And it would be interesting to see if you could actually take this hash map kind of implementation and make it faster than Bowtie, or at least make it faster than Bowtie per megabyte, because that would actually have some real implications to it. And finally, just the challenges of using 3 billion base pairs becomes really significant. I found some random bugs where just some of my assertions were being thrown off because, I know when you do floating points, you're not supposed to compare them directly, but I was getting differences above even one, there's at least 2. Division was having a large amount of error. Some functions need to be rewritten using some of the standard C functions like fread and ftell that you used to get the size of a file doesn't work over 2 gigabytes. You need to use different functions. Compiling for 64 bits, not that big of a deal, but doing these indexes takes a significant amount of time. And so doing all of those comparisons took several days to run all of these things. I'm sure a lot of people are running into that anyway. And it's a good amount of data to be collected, so you want to make sure that you're not screwing things up. And that's it. So if you want to get some of these slides, you can get them here. I also have this kind of an old CUDA card, but I got it off eBay for like $100, but it's got like 4 gigs of RAM. If anyone wants to use it, send me an email, and you can try it out. That's it.
Okay, any questions? Okay, so yeah, why don't we go to the next one?